Second promotory growth regulator is gibberellin. Actually, roots towards the Japanese history. So, in Japan, there was a time scientists are, while walking through the rice plants, they observed that some plants are surprisingly, some of the plants grow unusual taller, pale, lean, and thin and non-harvestable. These scientists are doubted those rice plants genetically dwarf got infected by the fungus and grown much taller than others, that those certain rice plants infected by the particular type of fungus is known as gibberella. This disease is called bacane or foolishing seedling. This made a confusion with the scientists to prove that gibberellic acids are responsible for this growth. It contains gibbon system. Gibberella fusicori. That extract is named as gibberellin. After that, many scientists are discovered more than 140 gibberellins denoted as a GA1, GA2, GA3, GA4, GA5, GA6 and so on. Among these, GA3 is a very common and well studied gibberellin. GA1 directly responsible for the stem elongation in plants. Recently discovered immature precursor which gets converted into GA20, which is precursor to the GA1, gets converted into GA1 through LG gene. This process was discovered to be catalyzed by an enzyme coded by LG gene. GA3 formula is C19H26O6. The precursor of the gibberellin is acetyl CoA. Reproductive parts have high concentration of the gibberellins. In 1912, Sawada thought that this disease caused by due to some substance secreted by this fungus. This was experimentally confirmed by the Kurosawa in 1926. In 1938, Yabuta and Sumiki were first to extract a crystalline substance from the fungus which they named as gibberellin. In 1961, Cross and his co-workers isolated 6 gibberellins from the fungus Gibberella, which is identified as GA1, GA2, GA3, GA4, GA7 and GA9. In the same year, Macmillan and his co-workers isolated the 3 gibberellins from the bean seed, which they identified as a GA5, GA6 and GA8. Gibberellins are present in all type of plants, that is algae to angiosperms, but rarely fungi and bacteria. They may be synthesized at a place where they needed, or they may be transported to the other places. The transport is done through xylem as also phloem. Their transport is non-polar. Transport is done through the xylem as also phloem. Their transport is non-polar. The sites of the gibberellin synthesis is young leaves. Anti-gibberellins. These chemicals are inhibit the gibberellins activity That is example phosphine D, B995, malic hydrazide. Physiological effect of the gibberellins. Gibberellin induces the stem elongation. Gibberellin induces the internodal stem elongation. When we treated with the gibberellins, the lettuce plant become vine-like. It has been confirmed on several plants such as peas, bean, tomato, pepper, sweet corn, summer squash, cucumber, cabbage. Genetic dwarfism. In genetically dwarf plants, the length of internodes is very much reduced, but the internodes remain same. When the gibberellins are applied to the single gene mutant of Vicia fabia and Pisum sativum, they become normally tall. The rosette of sugar beet can indicate extreme dwarfism. By the application of these gibberellins, Lang in 1956 observed a rapid growth or bolting on them. This similar behavior had been observed in dwarf rice, maize, and watermelon, squash, and cucumber. Substituting the cold treatment, the biennial form their vegetative body in the first year, then they pass through the winter season, and then they produce the fruits and flowers in second year. By exogenous application of gibberellins, many biennials can be induced to behave as annuals, and they no more requirement for natural chilling treatment for their flowering. Breaking dormancy. The exogenous application of the gibberellins has been shown to be capable of breaking dormancy of potato tubers and bursts of trees in winter. The exogenous application of the gibberellins induces the parthenocarpic fruits. Gibberellins are considered to be more efficient than the auxins. They are also applied to the stigma in a lanolin paste, increased fruit size and flower size. By the application of gibberellins, the size of the flower in geranium and camellia increased. Similar application of gibberellin also increased the size of fruit of vitis. Rapid maturity. Application of gibberellins in conifers shows rapid growth. Thus, it is used to economically important seeds. Elongation. In sugarcane, gibberellin stimulates the elongation of the stem. Sex expression. Gibberellins found to cause the maleness in some plants. In this function, it is antagonist to the auxins. Auxins causes the femaleness in some plants. Pamolin is a combination of the gene is a combination of GA4, GA7, cytokinin, 6-benzyl adenine. It acts as a apple enhancer. Malting. In gibberellins are used in beverages. It increases the malt production in barley grains by enhancing the starch digestion. Increase the fresh weight of patches and hay crafts. In storage of oranges, gibberellin prevents the rind disorder by delaying senescence. Gibberellin delay the senescence, thus the fruits can be left on the tree longer so as to extend the market period.